Welcome to Medical Grand Rounds. My name is Monica Farley, and I am the Director of the Division of Infectious Diseases, and this is one of our infectious disease slots for Grand Rounds. It's a pleasure for me to introduce a tag team. We have two people who are going to be presenting today. Dr. Andrew Neish, who is going to start us out. He's a professor of pathology and is an expert in gut microbiota and epithelial pathobiology, and he's going to start us out on this topic that will deal with the microbiome and its clinical ramifications by giving us a foundation of understanding of the basics of the microbiota and the science of it. He will then be followed by Colleen Kraft. Dr. Colleen Kraft is an associate professor in the Division of Infectious Disease in the Department of Medicine, as well as in the Department of Pathology. She's also the medical director of the microbiology lab here at Emory University Hospital. And she, along with Dr. Tanya Deere from GI, really run the Fecal Microbiota Transplantation program here. And she will take and transition to looking at some of the clinical applications of our growing understanding of the importance of a healthy microbiome, both in treatment of disease, but really fascinating in potentially preventing infectious diseases as well. So let me welcome Dr. Neish.
Well, thank you for that introduction. What I wanted to do today is talk about the microbiota, and what I'm going to do in the first 20 minutes of this talk is give some basic science background on this, about why the microbiota is important. Colleen and I have had these conversations over the years about what are the potential clinical utilities of this and ramifications. And I'm also going to do a single vignette from my lab about some of the role from recent data about what the role of the microbiota is and how it can influence health and disease. So my part of the talk is new thinking on epithelial microbiota interactions. And as Monica mentioned, I'm a professor of pathology, so that's my background is anatomic pathology. And I've always been interested in the tissue effects of, I actually started out in infectious disease looking at the tissue effects of enteric pathogens such as Salmonella, Yersinia, and such. And then transitioned about 10 years ago to looking at the effects of the microbiota, which, you think about it, is a far more common phenomenon than the very rare interaction with a pathogen. So to begin with, here's the microbiota, and this is a slide from a recent review. This famous line, there's 10 to the 14th organisms, mostly in the colon, although it's distributed through many tissues in the body. There's a skin microbiota, oral, genitourinary. Apparently there's some microbiota in the airway and some other tissues. There's 10 of the, so there's, the other point is supposedly there's 10 times more prokaryotic organisms in our bodies than there are eukaryotic ones. That's can be modified depending on how you count. There's also apparently 100 times more microbial genomes in our guts alone than in nuclei of our cells, implying there's a lot of biochemical functions that could potentially be meted by the microbiota. The microbiota is acquired at birth, and with the basis of modern sequencing methods, we're able to do a lot of the taxonomy and determine who and what they are. Go immediately to this slide. The acquisition of the microbiota is a fascinating concept. All mammals are born sterile, or at least theoretically, and there's some evidence, highly controversial, that there's some colonization of microbiota very early on during delivery and even in the placenta. But essentially, the microbiota we're born sterile, and there's a rapid acquisition of the microbiota instantaneously at birth. A lot of interest now in the difference between vaginal and C-section delivery, where the normal occupants, the lactobacilli in the vaginal canal, theoretically or likely play a role in subsequent immune and epithelial development, and the potential consequences of not having the proper microbiota. So that's an area of great interest now in the OB field. Anyhow, in mammals, they're going on a milk diet. This fascinating concept that a certain percentage of the proteins within milk is actually for the benefit of bifidobacteria and other bacteria, not for the infant, or at least in the synergistic sense it is. But it illustrates the co-evolved and intimate interrelationship between our microbiota and the host, being ourselves. And this changes over time with the introduction of solid food, and get great diversification depending on your diet. And in adulthood, there's lots of variations and lots of studies over the years characterizing associated taxonomic community structure of the microbiota in various disorders. And let me back up. And this brings up the concept of what is a dysbiotic microbiota. So the first caveat I'm going to say is most of these studies are looking at fecal material, and we'll talk about that. It's understandable because that's what you can get. It's a lot easier to sample the feces than it is to do a biopsy. It's easier to sample blood than it is to get a hepatic biopsy. But you got to remember that, and we'll talk about this. There's a microecology of the microbiota. But nonetheless, based on these fecal studies, these taxonomic association studies, there's a lot of interest in what's called a dysbiotic microbiota. And this is a concept that you hear a lot. No one really knows what it means, but it simply means it's dysfunctional, something wrong with it, and has an association with some disease state. It can be secondary to genetics. A classic example is NOD2 mutations that predispose to Crohn's inflammation, that form of IBD. Diet associated, a lot of interest in excessive or insufficient antigen or microbial load in our diets, and thus resulting in a maladaptive immune system. Antibiotic use is a very interesting. Martin Blaser is popularized this idea that antibiotic use can suppress normal growth of the microbiota and a lot of subsequent effects in health later on. And this is associated with a wide and expanding range of human diseases, and basically anyone in medicine can find their favorite disease that can be influenced by microbiota. The classic ones are IBD. That's one of the first characterized notions, and indeed the detection of the NOD mutations, which is a pattern recognition receptor, sort of catalyzed the whole notion about changes in the microbiota in inflammatory disease pathogenesis. And mucosal immunity all have major roles with the microbiota. There's effects of the microbiota in the CNS. We're interested in effects on Parkinson's disease, development of the immune system, effects on viral pathogenesis, response to antibodies, obesity, metabolic syndrome, a lot of endocrinologic abnormalities, autoimmune diseases, osteoporosis, and liver disease is another area we're very interested in. So these are all areas where, quote, dysbiotic microbiota can affect disease states. But usually the microbiota is symbiotic. So the mutualistic effects of the microbiota, these have been cataloged over the years. Competitive exclusions of pathogens. This is colonization resistance, and as Colleen will jump into, this is the major topic of microbiota occupying binding sites or utilizing nutrients otherwise used by a pathobiont or, say, C. diff, for example. The microbiota is known as metabolic, nutritional, and energy utilization, bile acid metabolism, a conjugation of various cholic acid derivatives. That process is very dependent on the microbiota. Short-chain fatty acids, butyrates, is a major energy source, and it's thought that specific bacteria with greater or lesser ability to extract these nutrients can contribute to obesity and other regulatory or metabolic effects. Huge amount of interest in adaptive immune regulation. The segmented filamentous bacteria and their putative specific role in controlling Th17 development and all sorts of adaptive immune regulation. And areas my lab is interested in is innate immune regulation, suppression of NF-kappa B, and tolerance of inflammatory signaling. Cytoprotective effects, the ability to induce patterns of genes that render cells less susceptible to subsequent insults. And what I'll touch base on as an example is epithelial development and the stimulation of barrier proliferation and restitution. But a little bit more background on microbiota. As I mentioned, the whole idea is a lot of these association studies are based on the idea of looking at fecal material, which, again, it is valid. But bear in mind, that's just the end result of the microbiota. The microbiota is a very diverse ecosystem. This is a schematic of the alimentary tract. The density of microbiota increases as you go oral to aboral. Essentially, there's not much in the stomach. There's only a handful. This is where your pancreatic juice, bile ducts comes in. The small bowel has Paneth cells and antimicrobial peptide that suppress microbial growth. There's not a lot of bacteria here, but then again, this is where the GALT is, the immune tissue. So this is where most of our recognition of microbial invaders or beneficial bacteria comes from. The colon here is a much more diverse and numerically vast, by orders of magnitude. Essentially, the cecum is a fermentation chamber. So there's a big change along the oral to aboral extent of the gut, and also in cross-section. There's what's the concept of planktonic microbiota, that's just floating through, presumably has some biochemical function, and those that are actually adherent to the epithelia itself, which may have more trophic effects on the epithelia and may have more role in blocking pathogen attachment. As you can see, again, bacterial load changes across the distance. pH changes as you get more fermentation and more of these acidic products. Antimicrobials go down, presumably it's why the numbers go up in the distal part. And oxygen changes. Even at the micro level, there's geographic changes in the microbiota. This is a schematic of the small bowel and the colon. There's a steep oxygen gradient. This is where your capillary and the intestinal villi would be, so there's less oxygen. a thick mucus layer, lots of Panna cells, antimicrobial peptides. And especially in the colon, there's this concept of a thin outer, loose, and a thick a mucus layer. And this is where a lot of the action occurs when we're talking about adherent pathogens and C. diff and these biolayers. The inner mucin layer is free of bacteria, whereas the outer mucin layer actually supports a whole population of mucosal-associated bacteria. They essentially use the mucin as an energy source. So the concept here is just to bear in mind when people talk about associations of microbiota, what are they actually talking about? Are these fecal samples, like in the environmental sense, or are these derived from biopsy samples or from various tissues, or and for that matter, when and where? So the variation of the microbiota is vast. It's an ecological system. So just to go into a vignette of some work in my lab. So this is the interface we're talking about. In pathology, here's an H&E stain section. This is the nice happy colon with a dense mat of bacteria over it. These are the luminal contents. And to me, this has always been a remarkable phenomenon that it tolerates this literal contact. But the other thing, this is a little bit of artifactual because it's informal and fixed. The traditional pathology doesn't preserve the mucin layer. And to look at actually what's going on, you have to go to different fixatives such as carnoids. And this is what you see when you do this type of experiment. And this is all mouse tissue. So what we have here is essentially the same area. Here's the colon. These are the nuclei of the colon. The green stain is mucin. That's the secretory mucin. Here's the thin layer, which sort of mirrors this, but here it's preserved. And this is this insulation that prevents the actual contact of the microbiota to the epithelia. And up here's the loose area, and what we also have here is a FISH probe, fluorescence in situ hybridization for 16S ribosomal DNA, or rDNA, which is a probe we can use to identify bacteria either en masse or specifically. But you can see up in the mucin, you have the bacteria, they're grazing up here in the outer mucin layer, but not entering the inner mucin layer. So how do we study this? One thing I want to bring up right now is my colleague, Renault Jones, who's in the audience, has established a germ-free notobiotic facility over in HSRB. And this enables us to study the biology of a mouse in a germ-free situation, such that there's no microbiota at all, or alternatively in a controlled situation where very specific forms of microbiota, either complex or simple forms, can be tested, and we can ascertain the effects on epithelial biology, immunology, and what have you. So I'll be talking about this in a second. Here's an example of an observation from a colleague of mine at WashU, Thad Stappenbeck, noticed one of the effects of the microbiota on intestinal injury, and this is the example I'm going to use. In a conventional mouse, here's the colon, or the colonic mucosa. It shows normal proliferation with some of the standard markers of proliferation. If you irritate the colon with what's called DSS, which is a chemical irritant, it causes intestinal damage. Once you resolve that or stop that treatment, you get a rapid healing response, and this is what it looks like histologically. And this is hyperproliferation, this open up space, that's edema. It's basically swelling and restitution. So this is a classic injury and response model. But what was in a remarkable observation, if you take a germ-free animal, you can provoke injury, but you get much, much less restitution. You end up with a very atrophic epithelia. And this brings up the concept that the microbiota has a role in restitution, or for that matter, in the normal physiology of the GI tract, in the mechanics of replacement of the epithelia. And to the GI people, this is in the audience, this is well-known stuff. But the epithelia has crypts. There's proliferation of stem cells. They migrate in an escalator up to the surface, and they're shed. This is a process that renews itself over the course of about a week in humans. There's also differentiation into the various absorptive secretory cells in the gut. So the intestinal epithelium is the most dynamically renewing tissue in mammals. Bacterial colonization is essential for normal tissue development and renewal, but the underlying mechanisms by which they modulate this is unknown. So I just want to show a quick series of experiments that illustrates the effects of the microbiota in this process. One of the systems we use is looking for wound healing. This is a cartoon showing the epithelia with a wound. Get dedifferentiation of the epithelia migration for healing. We can study this in vitro with the simple cultured cells in a scratch wound and observe the cell biology. Or more relevant in the medical sense is a wounding model. So what we have is a small animal endoscope. It's designed to take biopsies. It will take biopsies on a mouse colon. This is what this is. So this is a cut through the mucosa, and with the early healing, this is the neutrophils. And this is a system we use to evaluate effects of the microbiota on wound healing. So here's the biopsy forceps. And these are experiments performed by Ashfaq Alam in my lab. What he did was use the biopsy forceps to inflict wounds and observe healing over time. It takes about a week for these wounds to heal. There's no other underlying injury to the mouse. Here's a histologic image of the wound. You can see at day 2 the influx of inflammatory cells. But we subjected these type of sections to the analysis I was talking about earlier with 16S ribosomal probes to detect bacteria as well as the mucins. And Ashbach knows something very, very interesting. As I talked about earlier, in the normal epithelia, here's the surface of the epithelia, and there's this gap here. This is the mucin layer. It's lost here, but processing, that keeps the microbiota out of direct contact. Fine. But in a wound bed, when you're getting re-epithelialization, something very striking is the microbiota actually makes, as you can see here, actual physical contact with the epithelia. So this is remarkable and not found in normal biology, or at least in the adult healthy gut. So it brought up the idea that there's a different microbiota here and what would be potentially present on resolving epithelia. So what Ashbach did was a microbiota analysis of stage wounds. We got a mouse, inflicted these biopsy wounds, and prepared DNA after sacrificing the mice and dissecting out the wound beds. Prepared DNA from luminal contents, the wound bed, intact mucosa at days 0, 1, 2, 4, and 6 days. So here's the schematic. This section here was taken, the adjacent control, and then the luminal control. So what I'm going to talk about, and Colleen's going to talk extensively about microbiota analysis. So I'm going to go through this flow chart, which came from a relatively recent review, which I think is very instructive. It talks about when we analyze microbiota, what we're talking about. So here's the point. Here's a human microbiome sample. Usually this is, as I mentioned, a piece of fecal material. In other workers, this could be soil, could really be anything. In our case, it's actual biopsy. But here's the sample. If we're extracting DNA, there's 3 things we can do with it. The most common one is basically what we're going to be talking about today is 16S ribosomal sequencing. So the ribosome has variable in prokaryotes, has variable and constant regions. So you can make PCR primers across the constant regions and amplify up everything. But when you sequence across it, you get the sequencing reads from the variable regions. Therefore, you can use that to identify the taxonomy of the bacteria. And that's sort of the technique that revolutionized the analysis of the microbiota. And this technique and the subsequent software enables us to look en masse at the taxonomy of the bacteria. So again, compare sequencing of the ribosomal databases in bacteria, and that tells you it's a taxonomic description of the community structure. What organisms are present and what's their relative abundance. You can do similar stuff with 18S and look at fungi, and then you can do total, where the field's going now, total DNA sequencing, where you're sequencing everything en masse, not just by PCR amplification. Compare sequences. Here you're actually getting not transcripts, but the actual genes present there, and you can get more detailed analysis of who's there and some idea of what they're doing because of the gene content. Now, of course, you want to know more biologically relevant activities. You got to extract RNA, and this is traditional expression profiling by RNA-seq or whatever the current technique is. But from a sample, you can tell to try to determine what are the appropriate transcripts, both host and microbial, and get an idea about gene activity. And this starts giving you an idea what are the functions of the community, what enzymes are being present, what metabolites are they producing, what are they capable of breaking down. And this gets more into the functional analysis of the microbiota. Finally, we can get into extracting small molecules. This is proteomics, mass spec instead of a sequence-based analysis, compare references, you get metabolites. And this is metabolomics, and again, this tells you more about the functions of the community. So these, between DNA, RNA sequencing, and proteomic approaches, these are the various angles for looking at the effects of the microbiota in a biologic sample. So anyhow, what do we find with this system? So this is all sequence-based. So we have just two brief images here. This is called a stacked box plot. So the output of all the sequencing tells us the categorizing of the various species at the phyla or genus level, depending on what you're looking at. And what you can see is it changes over time, between day 0, 2, 4, and 6. There's some changes. Another way of looking at this is called a PCA plot, and we'll discuss this a little bit further with Colleen's data. This is a statistical method that clusters these into similarities. So what you can see is these are individual samples of intact mucosa. The software clusters and says that the microbial structure of all these intact samples is similar. Well, of course it is. It's intact. If you look at luminal contents, this is analogous to looking at fecal material. It's very, very different. It's the planktonic microbiota. It's very different than what's intact, attached to the mucosa, but it's similar to itself. It's flowing on by. So this is well known. There's planktonic and mucosal associated. But what we found, in addition to that, at day 2, a completely different population emerged at day 2. Then at day 4, it changed a little bit different, but it's vectoring towards the intact mucosa at day 6, and then ultimately it restores itself. So a population emerges in the wound bed and generally moves back. So this is classical ecological succession. You have a mucosa, you have an injury, you've changed the environment, you get a different population that moves in. For example, this is like a forest fire, like at North Georgia. Instead of the trees are all gone, here you're getting grasses and mosses coming in, then you get saplings and shrubs, and then eventually you get restoration of the forest. So anyhow, this is an example of changing community structure in a pathologically relevant context. So just some more confirmation, we can get the identity, what these things are. One of the main findings is Akkermansia and Prevotella are 2 taxons that seem to be associated. Lactobacillus comes up later. These are all organisms we can culture, and we can do some add-back experiments, and it's a topic of a lot of interest in our lab. And we can validate, say, for the example of Akkermansia, it's present in the wound beds by these hybridization-based techniques, but generally is quite rare, although present in the loose mucus layer. And then just, as I mentioned, these things have functional properties. We can do add-back probiotic experiments with this wound-associated bacteria, whether it's lactobacilli or Akkermansia. In wound beds, it can stimulate activation of ERK phosphate, or ERK MAP kinase, implying that there's some signaling events by these beneficial bacteria. We're able to show stimulation of proliferation within the crypts. So these are functional outputs of these bacteria that have been selected to be enriched in wound beds. And then finally, in a add-back experiment, these are the wound beds observed through the mouse endoscope. We can capture images sequentially and use image analysis software to quantify the rate of healing. We can do add-back with these wound-associated bacteria and show that they can accelerate wound healing. And we've shown this is through mediation of proliferation of the epithelia and actual migration at the cell biologic level. So basically, wound-associated microbiota, it's a subset of normal microbiota, including Akkermansia and lactobacilli. They expand in local microenvironments. So again, it brings up the concept is the microbiota is very dynamic. This is a very defined injury. In something complex like IBD, it's going to be very complex because you have areas of activity, restitution, denudation, or normal appearing mucosa. So it's going to be very complicated. We're not getting the redox-dependent aspects. There's some signaling mechanisms we've been working on. But these are possible mediators of the beneficial effects of the microbiota. And the conclusion suggests an intriguing role for specific intestinal bacteria in the modulation of cellular signaling pathways. It's a likely mechanism for commensal-mediated homeostasis and restitution and possible therapeutic utility. And I'll leave it at that. This work was done by my lab. Ashfaq Alam did the wounding models. Renald Jones, of course, has the germ-free facility, and other members of my lab who contribute a lot of this. So at this point, I'm going to hand the background we discuss off to Colleen, who's going to talk about more of the practical applications and what can be done in the clinical realm with this topic. Thank you.
Thank you. And one of the great benefits of working with Andy Neish and Renald Jones in that collaboration and just having discussions with them is to think about things in mice. You can get very mechanistic and very specific and do very controlled experiments. And I think one of the things that is beneficial about working with basic science in general as a clinician is to understand some of that from a real basic level and then try to see how we can apply it in people, the questions that we can ask and the direction that we can go based on these observations that Andy and Renald have made. Now you're going to be moving to the Times New Roman font of the presentation. This is the more clinical application, like Andy already mentioned. And so I wanted to start out by just a question of what do we use to manipulate our microbiome? So Andy talked a little bit about dysbiosis, and we're definitely going to get into that. But we use lots of different things in theory to manipulate the microbiome. Whether or not these all have efficacy kind of remains to be seen. So prebiotics, I had to Google this because I kind of forgot what they were. This is sort of dietary things. It can be some environmental things. And this really has some non-digestible fiber that provides some of that fermentation that Andy was talking about in the colon. And it can include those things listed up there, except that they all have to be raw. So enjoy that dandelion green salad that you'll eat today. Probiotics are obviously something we talk and think a lot about. I would say I get asked about this every day by somebody. Do they work? Should we use them? Should we use them in our patients? So this is a therapeutic supplementation with exogenous viable bacteria. And there are things in mice especially that have been shown to modulate inflammation, augment barrier function, and stimulate those reparative responses that Andy had mentioned in his part of the talk. And of course, near and dear to my heart is fecal microbiota transplantation. And this has shown to be successful in C. difficile colitis. And right here you see one of our famous donors who agreed to be interviewed for a website, so about how he would donate his stool, one of the pathology residents. So whether or not we thought this was ever going to happen, when I was an ID fellow way back in the day, just kidding, we would joke about this. There was actually an up-to-date card that we would look up about it, but nobody actually did this. And then all of a sudden, the paradigm really changed. And so it became more mainstream. And it's called lots of different things. Cliff McDonald at the CDC, who has also been a mentor to me, really never wanted me to use fecal, so he tried to get me to write intestinal microbiota transplant and everything. No journal would ever accept it. They made me switch it to fecal because that's sort of the standard nomenclature. Those clever Canadians like Microbial Ecosystems Therapeutics, which I think sounds a lot nicer than fecal transplant. And this is really, by definition, just taking someone's stool from a healthy donor and infusing a liquid suspension of this in order to restore a normal microbiota. I think one of the things that really accelerated this as a clinical use was this New England Journal trial that happened in January of 2013. There have been 2 since then, so there's not a lot. The holy grail of medicine is usually a randomized controlled trial, so this is really what I think pushed us into more and more people doing this. In this case, they used an oral vancomycin regimen, which really is our standard of care, followed by nothing, so the standard of care, just vancomycin, bowel lavage, just to make sure that giving somebody Miralax or not Miralax, Go Lightly, didn't actually help your C. diff on its own, and then bowel lavage with donor infusion. And interestingly, this study was considered superior, and they were doing it by duodenal infusion. So most of us don't doing that, most of us meaning people in GI, not ID, most of us don't infuse this by upper endoscopy. We usually use colonoscopy. But anyways, this was stopped early because of efficacy in that infusion population with minimal adverse events, and they showed by 16S a restoration of the microbiota diversity. So our program started here in July of 2012. And it was stuttering along probably until that first paper. I will also say it was stuttering along until I partnered with Tanvi Deer, who essentially has created the storefront for this clinical program in the GI clinic. She has she usually performs it via colonoscopy, and sometimes we do them inpatient, just obviously depending on where the patient is. We've treated nearly 300 people with fecal transplant with greater than 95% success. Sometimes people require multiple, depending on how well they do after the first one, but most of our people do better. We also use, which is different than many programs and why Tanvi and I work together, so most of the places, the GI suite kind of does it all. But here, I coordinate and mix the stool, so I'm the poop mixer. And we coordinate local donors, like the one I showed you earlier, and I personally mix the stool, which is then transplanted. So, and you may or may not know there are some donors sitting in the audience, so look at your neighbor with concern. They've washed their hands, don't worry. So one of the exciting things about being in an academic setting is being able to ask questions when you're taking good care of patients. And so these are some of the projects that have stemmed just from some internal funding from Emory and actually also from the CDC on some projects that we've asked some specific questions in our fecal transplant program. So I'm going to be talking a little bit about the first one, which is the microbiome of long-term acute care patients. And sorry, I got Fitbit, can't get the time. The microbiome, Emory University Research Committee has funded myself and Virginia Schaefer. The question that I asked was the question that Andy Neish had pointed to during his talk, which is is stool representative of the microbiota, of the luminal microbiota? So with Tanvi Deer, we did, we meaning her, she did the biopsies. We did biopsies at the before and after FMT to see if the luminal microbiota was similar to the microbiota of the stool. And Tanvi Deer also received a seed grant to look at fecal transplant after the first episode. So trying to move, like, should the standard of care be after the first episode instead of after medically refractory C. difficile? And Virginia's University Research Committee grant had to do with the use of fecal transplant in patients with ulcerative colitis that now have J pouches because they've had their entire colon and rectum removed, and to see if, rather than using VSL3, which is a probiotic, if using fecal transplant would kind of basically fix their pouchitis, basically trying to make that ileum that's become a pouch to make that look more like a colonic mucosa. We've also had the benefit of just asking some questions just in a retrospective way. We have been a part of the literature that is reporting adverse events after a fecal transplant. We had a lady that ended up with diverticulitis after our FMT. So all of that work is trying to support and show, the types of adverse events that patients can have after these types of therapies in order for, that all to kind of be known as a clinical safety profile. We've also looked at whether individuals who are immunocompromised, if it's effective and safe in those individuals from a retrospective chart review, and also looking in older adults, which was an idea I had while I was mixing stool. I get a lot of thinking done while mixing stool, so I thought we should look in geriatric population because they have they are at high risk for C. diff. So hopefully you all know this by now, but this has sort of been my mantra for the last 4 years, is how does this actually work, and what are we learning about how it works? So all of us hopefully are at a homeostasis, but you certainly know when you are not at a homeostasis, when you have transient dysbiosis. And you may have it from lots of reasons. You got rotavirus from your kid, you took antibiotics and you're just having some dysbiosis after that. You may also have it from traveling. So you may have a transient dysbiosis. There may be an expansion back to this to sort of a homeostatic gut. And we sort of even intuitively kind of know that about ourselves. However, if you take antibiotics, it causes potentially, and we'll talk about this a little bit more, a diminishing of the diversity of your gut, which allows it to be able to have C. diff as an overgrowth. So if you have C. diff exposure plus that antibiotic exposure, you could end up with persistent dysbiosis, and you can end up with very chronic dysbiosis as well. We all have had those patients that have had multiple episodes of C. diff and can't really seem to get better. And that is what fecal transplant, called here bacteria therapy, really is to do, which is to disrupt that dysbiosis that they can't get out of and to clear the C. diff, or at least put it back into its niche and have it come back to homeostasis. And those who have heard me speak, their eyes will roll back into their heads when they hear the garden analogy, which I say to patients and providers, which is our gut is like a garden. And when we use antibiotics, we're killing all those fruits and vegetables. They're getting killed off. And C. diff kind of grows up like a weed, might have been there colonizing you, or you might have picked it up from the environment. And we then use vancomycin, fidaxomicin, Flagyl to kill those weeds. So we're giving it a weed killer, but we're not replanting the garden. So you can imagine how somebody can enter that cycle of dysbiosis by having repeated weed killers given to their garden, but not replanting. I think one of the most exciting things that we've learned in this work is that it may not be, and this is where probiotics and the idea of having a really good probiotic comes in, maybe we don't have to replant the whole garden using all those seeds. Maybe there's just a few seeds, which is an ecology term, a keystone species, that are required that then let all your other plants come back in. So they kind of create that environmental ecologic niche in your gut that then allows all the E. coli and everything else to kind of get back into to shape. Here are the first poop pills we gave at Emory Hospital. So another benefit of, again, treating patients with fecal transplant is we've also been able to be on the cutting edge of some industry trials where they've been making these pills. And as I've already sort of described to you before, the keystone species, at least by this company, they believe that the keystone species would be the spore-forming gram-positive bacilli, which actually kind of include Clostridium, interestingly. And so is the niche that needs to replant that garden and allow that garden to be to kind of grow again, is that just the spore-forming organisms? The way that they created this pill is they started with stool, shocker, and then they treated it with alcohol, so they basically distilled it, and that killed all the vegetative bacteria. And so what they then did was they counted the spores by like a fluorometric method, and they put into these pills a certain number of spores. And they actually got to the point where they kind of knew who their high spore donors were and low spore donors. So they kind of get the high spore donors to give a lot more so they don't have to distill as much. So these were our first pills that we gave here. And Cindy in the back was actually part of that study, not as a participant, sorry, as a study coordinator. So the results of this study were published in the Journal of Infectious Diseases, and we were really excited about it. In all of these, this is kind of a weird graph, but it's dissimilarity. So you want your dissimilarity to the donor to go down. So you can see in all four of these time points here, in groups, all of them went from being dissimilar to the donor to being similar. And so it's kind of a weird way to present the data, I agree. But basically, there was efficacy, and their microbiota sort of restored to near the donor. So we are just finishing up the phase 2, and unfortunately, this failed to meet its goal in the phase 2 trial. So a lot of people had a lot of hope for this type of product. But again, they're still trying to figure out what exactly happened, if it's something about the pills, the preparation. So this was a pretty significant disappointment. But again, this shows where we are with this type of research and kind of where we're going. So what I want to finish talking with you about is really about antibiotics and multi-drug resistant organisms. So I credit this slide to Caitlin, who works with me. She did all of this work, all this nice figures here. But we use antibiotics to try to help treat infections. I mean, they're a necessary evil, like the title says. But we end up with dysbiosis, which I've kind of mentioned already, maybe a lack of healthy bacteria, maybe too much bad bacteria, and this is where things can, like C. diff, like VRE, can come in. And then the worst, it gets uglier when we have multi-drug resistant organisms involved. You can get clinical infections with those multi-drug resistant organisms. And then I like the right, which is the super ugly. So these are very serious infections. You can have multi-drug resistant organism infections, like I said, hospitalizations, really toxic antibiotic, and death. And my children, here they are, made this lovely Lego figure set of a fecal transplant for me. They were super excited when I came home. I won't go into depth because it's a little, if taken out of context, doesn't look very good. But that's me with the ponytail. And I never do this because I'm not a GI doctor, but anyways, the Kraft children have participated in this, and here they are. So our work with the CDC looking at the long-term acute care microbiota, so we did this at Wesley Woods down the street, and we looked at 10 patients that just had a C. diff test sent because they had diarrhea, and we looked at C. diff status, whether or not they had multi-drug resistant organisms that we could culture out of their stool, and looked at some other clinical metadata. And this is actually published in AGIC, if you want to look at it. But what I want to point out to you are these bar graphs, like Andy mentioned. And so we have our healthy donors. Actually, neither of them are in the room right now, but they have seen their own microbiota sequenced. So here are our healthy donors, and I really want to point out the percentage of Bacteroides. So as you're reading these papers and thinking about this, I think a healthy microbiota is really represented by a high percentage of Bacteroides, so 40 to 60%. Many of you in this room, maybe like me, thought that our guts were made of what? What did we think was in our gut? The most common bacteria in our gut? E. coli. So it turns out that E. coli and all those proteobacteria that we see as clinical infections are actually less than 1% of our gut. So our gut is really anaerobic, and you kind of wonder why we don't see more clinical infections from it, but that's another topic for another time. So you can see over here, this next patient, well, he or she looks like she's got plenty of Bacteroides. Well, it turns out this person was only receiving inhaled tobramycin. So they weren't really getting any systemic effects of that antibiotic. So their gut is still sort of preserved, if you want to call it. And see, over time you can see just some differences, but I want to really call you to the attention of something that's like monodominant. So this turns out to be mainly Pseudomonas in this person's gut. This is mainly Reichenelliaceae. And these ones in the yellow turn out to all be VRE. So at the point at which we can detect something that's abnormal, like a VRE swab, that is all you have in your gut, most likely. And I kind of call it the last man standing. So VRE, because of all of our antibiotics, we're not always giving things that treat VRE up front. And so that ends up being what's left. And so as we talk about how we characterize dysbiosis, I'm going to say a real easy way, a low-hanging fruit that we're hoping to expand upon, is that anybody that's VRE positive in their stool has dysbiosis. And the next question then, I think that we'll be talking about in the future state, is what are we going to do about that, and should we do anything about that in our patients? Eric Paymer is at Sloan Kettering. He's an infectious disease doc. He's actually coming to speak at Pathology Grand Rounds in September. He also showed this, very similar thing before we did, that basically these people had the last man standing. They had only VRE left, and 20 days later, this person's bacteremic with VRE. Same with this person. As their gut just becomes only VRE, these are all bone marrow transplant patients, they puts them at risk for that to be in their bloodstream. So the new frontier really, I think, is multi-drug resistant organism elimination. And like I already said, if we can have a clinical surrogate for dysbiosis and monodominance in our guts, this might be a place where we can intervene. Should we intervene? With what should we intervene? Those are all questions that remain to be answered. But that's when we talk about the prevention of infections. We talk about kind of futuristically, how can we make our patients' guts sort of go back to normal? Is that safe for them to go back to normal? So in Eric Paymer's work, he's working with people that are getting toxic chemotherapy. Should you be giving those people wild-type E. coli back in their guts after they have only VRE left? So these are all questions that are still sort of out there to be answered. So one of the things that we described here at Emory, but we kind of put tentatively in this paper, so Marshall Lyons on this manuscript, Rachel Friedman, we had two patients who were solid organ transplant recipients. They both received fecal transplants. And the renal transplant patient, who had been in the hospital pretty much every month for a year or more, she would go in the hospital for a urinary tract infection with a multi-drug resistant organism, as you can see here, so requiring IV antibiotics. Then she would be in the hospital the next month for C. diff. And she'd be in the hospital the next month for the urinary tract infection. It was literally an every other month thing. So eventually, she'd had enough C. diff that she could qualify for a fecal transplant. And we did the fecal transplant. And we also noticed, really anecdotally, that her urinary cultures became, first of all, it was no longer Pseudomonas, it was actually Klebsiella. And it was also pan-susceptible. So we were really impressed by that result. But we kind of just tentatively talked about it in our discussion. We didn't make much mention of it. But this sort of idea and result has been sort of now verified, again, just in case reports and small interventions, but that we can actually restore the gut microbiome, which may be causing these infections, sort of start it back over at a wild type, a normal homeostasis. This is another one of those descriptions where they had a patient that had both Clostridium difficile and VRE, which I hope you can appreciate makes sense in somebody that's really dysbiotic. It's gotten a lot of antibiotics, doesn't have much bacteria left, so all these MDROs are in there. And again, here's the donor, here's the recipient, and this is the recipient at the time of the fecal transplant, and then over time they sort of resemble the donor again with that Bacteroides fraction, then they kind of settle out on their own. But they also lose the enterococcus that had predominated in their gut completely. This is a review that Eric Paymer wrote in Science, just about using the intestinal microbiota to combat these antibiotic-resistant pathogens. And I think I've shown this to you over and over again, but I really want you to remember it from this talk, is that here's our pre-antibiotic diverse microbiota. Here's what happens when we get antibiotics. An expansion of the antibiotic-resistant bacterial species occurs, and then now we can maybe eliminate those resistant species by microbiota repair. And this is, again, the same thing, but very interesting way of looking at it. Just looking at antibiotic-resistant genes that can be detected by 16S in their, and whole genome sequencing in their stool. And in these individuals, here's the donor that has no antibiotic-resistant genes. Then before FMT, they have them. Post-FMT, they actually still have them, so these individuals got another one. And then after that, it sort of cleared that antibiotic resistance genes, and it went to look sort of more like the donor. So what's Emory doing about all of this? So we've sort of created a microbiota enrichment program. And we are looking, these are 4 studies that are investigator-initiated from Emory. We're also involved in 3 other industry clinical trials, but I haven't listed those up here. These are all ones that we're doing here. So we're looking at fecal transplant via these oral spore pills, like I've described. We're looking at fecal transplant in people, in renal transplant patients that have a multi-drug-resistant organism colonization after infection. So these people will undergo a fecal enema after they've had infection with VRE or CRE, and we'll be evaluating whether or not they can clear that resistance and restore the diversity of their microbiome. We're also doing 2 descriptive studies with the CDC looking at the impact of surgical prophylaxis. So we're going to take donors to renal transplant donors, and look at them because they're very healthy to donate a kidney. We're going to look at them before and after their gut microbiota, before and after they get cefazolin as their surgical prophylaxis. And then we're also looking at the microbiome related to international travel. So what can you do? You're sitting there, you really want to do something about gut microbiota and multi-drug resistant organisms. So the best thing you can do is avoid unnecessary antibiotics in your patients. I want everybody to raise their right hand, just kidding, and promise me. I thought about having this picture on every ward to say, you either need to get smart about antibiotics or you're headed for the poop cafe. So don't use unnecessary antibiotics in your patients. Think about what you're doing. Ideas of specialty exists as much to withdraw those antibiotics from your patients as much to tell you what to do. So please guard your patient's garden very carefully. If your patient has C. diff or other multi-drug resistant organisms, feel free to email me. And always happy to have participants to help us out with all these projects we have going. So, this was a title that Cliff McDonald put on an editorial he made, which was, "This is now the dawning of the microbiome remediation for addressing antibiotic resistance." So thank you, and Andy and I will take questions.
I can start out the questions, and this is really for both of you. Given what you told us about wound healing and the relationship with the microbiota and the idea of using FMT therapeutically, can you talk about the use or the applications in inflammatory bowel disease?
So I'm not a GI doctor, that's my first caveat, but in talking with Tanvi Deer, it's a much more difficult and complex environment than C. diff. And so I think there's potential benefit, but I think we're still trying to piece out in what stage there might be benefit. So is it when somebody is pretty quiescent that you introduce a new microbiota? Is it when they're really flaring? I think it's a much more complex immunologic-mediated scenario. There are about 30 clinical trials going on right now about fecal transplant and IBD, and a lot of that data hasn't really been published yet. So that's from the clinical standpoint.
Yeah, just to second that, the reason we went to that biopsy wounding is because it's synchronized. And in an IBD, as everybody knows, it's at various stages of activity. You may not want to put a bacterial preparation on a denuded epithelium, which is going to have access to this systemic circulation. On the other hand, it might be very beneficial in the sort of the late resolution stage. Add mix onto that the effects of steroids or whatever. It's very complex. But as Colleen mentioned, potentially as maintenance for remissions might be more reasonable. But again, it's a very complex situation and a lot of variation on that.
So, Andy, you talked about how some of the bacteria stay in the pulp. They're always there, not coming out. Those have potentially what is their effects because they're good at clearing and colonizing.
Well, there's a certain logic to that. And so the question is, are the bacteria that are adherent to the epithelia, are they more likely to have more benefits biologically? They probably have more role directly interacting with the epithelia, but they may not be the fermentative ones that are involved in breakdown of producing metabolites. They may have a very, very different function. So again, these are just, it's like asking what's more important in a forest, is the moss or the trees or the birds flying around. They'll have roles to play. But the ones that are epithelial associated presumably have evolved some type of pylon or attachment the way a pathogen does. It maintains that niche there, and thus we can assume that they've co-evolved for a longer period of time. It's just an assumption.
There it is. Thanks for that great talk. It was kind of interesting to live through some of this and see some of the side and unanticipated benefits of FMT. So my question is, is there something that we could do? So right now we're either doing it via colonoscopy. The patient that we published, or the two patients we published, we did it via nasogastric or nasoduodenal tube, and we mixed up a large amount of stool volume. How much of this is enough to actually restore what's needed? Could we get away with something that's smaller? Do we need to use the branded and copyrighted or patent-protected series stuff, or can we just mix in a little stool and just something palatable and throw it on down?
So a lot to unpack there, Marshall. And when you mean we mix the stool, you mean I mix the stool? Is that what you're saying? I think it's pretty clear we shouldn't be using the patented, Series Health, but it's not showing to work in the phase 2. So at a Freston conference I went to that was dedicated to the microbiota, I think for C. difficile, probably you don't need very much. So I think we usually do 250 cc's via colonoscopy, 10% in glycerol. 10% of that is glycerol, and then for nasogastric, which thankfully we don't do anymore because I had to do those. It's not enjoyable. We used about 100 cc's. And so the thought is that you probably even need a lot less, but nobody's really done a down dosing study where we've looked at that. For fear of sounding glib, I think because of our very clean society, it's possible that we could be having sort of auto fecal transplants that we're very clean and that's not happening. I'm not advocating that we have more feces around that we put our hands in, but I think people that they've shown in kids that parents don't wash the pacifiers, that they have more of a microbial diversity in their gut. If you don't use a dishwasher and you hand wash, all that grosses me out, actually. So anyways, I think the point I'm trying to make is that I think it could take very little. I think that my personal hope is that one of these pills will be able to be enough and be the right concentration. That's really a holy grail if you think about it, because those people will be making a lot of money, because we'll use it as sort of a chaser for antibiotics. Sorry. I don't have time to do that. But if somebody in this audience really wants to put it into little pills, be happy to help you out with that.
Have people thought about bypassing the whole stomach acid situation and going versus poultry?
No, we're doing enemas in a couple of our studies, but we haven't thought about suppository. That's a good one, actually.
Another comment along those lines, and Colleen and I discussed this, is to mention the terminal ileum is where the gall is, the gut-associated mucosal tissue, and basically all your mucosal immunity is in the upper GI tract. So NG is going to access systemic immunity and could have effects on IBD and autoimmunity. If you went by colonoscopy, you're just bypassing that entire system, and there would be no or much less effects on systemic immunity. So the delivery, no one's discussed this, but theoretically, there's a very strong reason to believe it'd be very, very different responses.
So the question by Dr. Tata is, we give patients, her renal transplant patients, alternating months of antibiotics. What happens to their intestinal microbiome? So let's look. This is what I think happens. So here it was. There's an antibiotic-depleted microbiota. You're going to get expansion over time, so they're going to get drug-resistant depending on what you're using. You do the alternating to hope that the population loses the resistance to one and doesn't, you're kind of trying to, but eventually there may be that population that takes root that's resistant to both of those antibiotics, and that just keeps a cycle. So we are looking at, we're going to be trying to enroll your patients in this study that we're doing to try to eliminate if they get to a point where they're multi-drug resistant organisms, to eliminate that from their microbiome.
This is a question from the VA, which kind of piggybacks off of that topic. So for patients who are high risk, like transplant patients, has there been any thought given to banking of stool prior to their inevitable insult and possible need for restoration in the future?
Yeah, you guys need to be in this field of research. You're asking all the right questions. So Eric Paymer, who I mentioned, who did this slide, actually, he is banking stool before those patients. So after the description of the bone marrow transplant patients that get VRE, after their induction chemotherapy and antibiotics, he now is auto-banking. So even though they have malignancy and they're not entirely well, the only way the FDA would let him do that is for him to auto-bank their stool. And then after they get induction and kind of wipe out their gut microbiota, they're randomizing who gets it back. And I just looked yesterday, and those results haven't been published. But the goal of that study is also to see if their engraftment is better. Looking at all the clinical variables to see if having a restored, which I call restored microbiome, or back to their normal, if that's actually really beneficial for the patients, even in their, malignancy treatment. So yes, people are doing it, but only in very small research studies.
Thank you.
Thank you.